What the study says.
So imagine walking into an exam room, right? You're bracing yourself for the usual routine, a blood draw, maybe a lumbar puncture.
Oh, the usual fun stuff.
Exactly. But instead, a golden retriever trots in, gives your shirt collar a quick sniff, and bam, correctly identifies a major neurological condition before you've shown a single tremor.
Yeah, I mean, it sounds like a complete comedy sketch.
It really does. But, as medical doctors combing through the latest clinical literature, we're tracking how this bizarre scenario is actually getting incredibly close to reality. Welcome back to What the Study Says. I'm Dr. May.
And I'm Dr. Kai.
And today we're bringing you this incredible new study published just this year looking at trained dogs detecting Parkinson's disease.
Right. Our mission for this deep dive is to really explore the potential of canine olfactory-assisted diagnosis. We want to look at how decoding what a dog smells might, fundamentally shape the future of early clinical screening for you.
Exactly. Because to understand how a dog could detect a brain disease, we have to look at the physical byproduct they're, actually smelling.
Yeah, so Parkinson's patients often present with this significant overproduction of sebum on their skin.
Which is like an oily, waxy secretion, right?
Spot on. And it's not just a random dermatological quirk. Parkinson's doesn't only affect motor function, it actually impacts the autonomic nervous system.
Oh, I see.
Yeah, so it throws oil production into overdrive long before the classic tremors ever even start.
It's honestly like smelling a burnt clutch from outside a car. I mean, the smell of the friction hits the air way before the transmission actually fails. So that altered sebum is basically the check engine light for Parkinson's.
That's a really great analogy. And what's fascinating is that this shift in scent was initially flagged by a human.
Wait, really? A human?
Yeah, Joy Melnin. She's a woman with a hypersensitive sense of smell, and she actually noticed her husband's scent change years before his formal diagnosis.
Wow. So if a human could detect that shift, it naturally made researchers wonder what a dog's nose could do. Because, their olfactory system is orders of magnitude stronger.
Exactly. They realized this altered sebum acts as a highly specific biomarker.
But when I read through the trial methodology, my immediate thought was contamination.
Oh, sure.
Like, if you're taking skin swabs from patients, how do you keep the dog from just smelling, the laundry detergent on their shirt or their specific diet instead of the actual Parkinson's biomarker? Because you can't exactly bring newly diagnosed patients into a kennel to test this.
No, definitely not. But they controlled for that through a really rigorous double-blind trial using dry skin swabs.
Okay, so how did they set that up?
Well, they took samples from 40 newly diagnosed Parkinson's patients who were entirely drug naive.
Right, ensuring medication wasn't confounding the scent.
Exactly. And they compared those to swabs from 50 healthy controls. Then they trained 2 dogs, a golden retriever and a lab cross, to sniff these swabs presented in lines of 4.
And crucially, to your point about isolation, neither the dog's handler nor the experimenters in the room knew which swab was which.
Right. It was completely double-blind.
It's almost unsettling to think a dog's nose can rival our laboratory equipment. Looking at the results, the dogs achieved between 70 and 80% sensitivity.
Which means they correctly identify the Parkinson swabs at a very high rate.
Yeah, but the specificity is what really caught my eye. It was between 90 and 98.3%.
Meaning they ignored the healthy controls almost every single time.
Exactly. That false positive rate is better than some of our standard clinical panels.
It is incredibly precise. But, when we look at numbers like this in the literature, the immediate question is always scaling.
For sure. Because we aren't going to be putting golden retrievers on the payroll to run hospital wings anytime soon.
No, definitely not.
So how do we actually operationalize this for your next physical? Like, what is the tangible impact here for clinical screening?
Well, the goal isn't to replace formal diagnostic tools with a canine unit. It's about leveraging canine olfaction as a rapid, non-invasive early screening tool to validate new laboratory tests.
Okay, so if a dog can consistently flag a specific swab, we can then run highly complex chemical analyses on that exact sample to isolate the specific molecular markers.
Precisely. Which opens the door for early medical intervention way before the motor symptoms degrade a patient's quality of life.
We essentially use the dogs as the ultimate gold standard to calibrate our diagnostic machines.
Yeah, because sebum production changes so early in the disease progression. Identifying that exact molecular target is the holy grail of screening.
And the blueprint for that detection technology is already perfectly engineered by nature in the dog's nose.
We just have to learn how to read it.
Which leaves us with a pretty provocative thought to wrap up on.
Oh.
Yeah, think about it. If a dog can smell the exact molecular signature of Parkinson's disease on a simple dry skin swab today, how far are we from engineering an electronic nose for every doctor's office?
Oh, wow. Imagine a small device on the desk that just sniffs a standard swab and gives you a readout in seconds.
Right. It would completely reframe how we approach early neurological detection.
It really would change everything.
Well, if you want to keep exploring the absolute cutting edge of clinical research with us, be sure to subscribe and share the channel, What the Study Says. Until next time, maybe pay a little more attention if a dog insists on sniffing your jacket. They might just be giving you a free checkup.